Hello everyone, my name is Josh and I'm a systems engineer here at Owlstone Medical. Myself and my colleague Riccardo will be presenting today on the development of portable breath analyzers here at Owlstone, with a particular focus on the OMED Health device. Owlstone Medical has a history of the development of products for academic research purposes, and the OMED Health breath analyzer is the first point-of-care device that we have developed. Today, we're planning to talk through the development of this product, with a focus on how our learning and experiments were used to optimize the product performance in the different aspects of technical development. The OMED Health Breath Analyzer project started around 3 years ago, with the aim to create a device that can be utilized at home by customers who are suffering with digestive health issues. Digestive health issues often require multiple GP and/or specialist visits in order to receive a diagnosis or treatment, so there is a significant market for an at-home solution that will allow patients to monitor their conditions and report these to a doctor in order to get help sooner. As part of the product development of the OMED Health Breath Analyzer, we determined the optimal sensors to utilize for the detection of on-breath hydrogen and methane that would fit the requirements of this device. We then designed the electromechanical aspects of the product through a rigorous prototyping process. Following product design, we knew that we would need to develop a matrix of sensors responses which can be used to train an algorithm to calculate the on-breath hydrogen and methane concentrations from the sensor responses in a sample collection. This required the design, building, and commissioning of a synthetic breath rig that could generate a test gas mixture consisting of a set of desired hydrogen and methane concentrations. Using this data, a machine learning model was built in order to evaluate these raw responses and output the calculated hydrogen and methane concentrations. In this presentation, I will discuss the design and development of the synthetic breath rig and the applications that it has, and following this, my colleague Rick will discuss the development and implementation of the machine learning model. The first thing we needed to do was understand the factors that affect the baselines and responses of the selected metal oxide sensors and how we can control them. Customers will be using this device worldwide and will be giving samples in a myriad of different environments, and so we needed to ensure that any algorithm solving for the on-breath gas concentrations would be able to compensate for this. As such, research was carried out to determine a window of ambient humidity and temperature values that would represent environments where these devices would be used. We then purchased an environmental test chamber that was capable of controlling the internal environment within this envelope. As well as this, we researched a range of concentrations of on-breath hydrogen and methane that we would expect to see in a region of pathological relevance. This allowed us to select supply gas concentrations, as well as design how the constituent test gases would be balanced. What all of this research allowed us to do was write a list of requirements defining the exact characteristics of what we needed a synthetic breath rig to deliver upon. The two graphs shown in this slide demonstrate the environmental conditions and gas concentrations that such data was collected with. On the left, we can see a graph of the ambient absolute humidity plotted against the ambient temperature. The curves demonstrate how the actual water burden increases with temperature with the same relative humidity. In this graph, points are labelled as both phase 1 and phase 2. This is because our initial focus on the training data collection was on regions where we would expect this device to be most likely used, i.e. in an indoor home or office environment. Phase 2 expands upon this to further detail environments representative of outdoor environments in both sunnier and colder climates. In the graph on the right, we can see a grid of hydrogen and methane concentrations. In our research, we found that we could typically expect to see concentrations of up to 75 ppm of methane and up to 100 ppm of hydrogen when collecting breath samples from users. We collected data in clusters across this concentration space and at multiple different environmental conditions. The machine learning model we would then be able to use this data to compensate for the environmental conditions the breath sample would be collected in. The graph on the right demonstrates just one of the many concentration clusters that we used during the training data collection experiments. Following all of the pre-research, we came up with this design for the synthetic breath rig delivery system, which is summarized in this detailed system diagram. A series of parallel thermodynamic mass flow controllers are set to each deliver a different constituent gas, be it nitrogen, oxygen, carbon dioxide, hydrogen, or methane. These gases are split into a humid fraction and a dry fraction. The humid gas section is humidified by passing this gas through a set of Drechsel bottles, and it mixes with the dry air fraction after this. By varying the ratio of these two fractions, we can control the relative humidity of the delivered test gas. The concentrations of hydrogen, methane, oxygen, and CO2 are varied by changing the ratios of the constituent gases controlled by the mass flow controllers relative to the total flow rate. We know what the concentrations of each gas are in the supply cylinders, which allows us to determine what each flow rate needs to be in order to reach the desired concentrations of the final test gas. The test gas comprised of a lower than ambient O2 concentration, offset by carbon dioxide, in order to represent human breath. At the outlet of the rig, we place our OMED devices under test. These are attached using a set of bespoke clamps, which allow for the test gas to pass directly through the device, as per a typical breath sample. In order to control the environmental conditions of the devices under test, they are placed in an environmental test chamber for the duration of the experiment. There is also an equilibration time at the start of each experiment to allow for the sensors to reach a stable baseline in the new environment. The performance of the environmental test chamber is monitored with a series of environmental sensors alongside an inline sensor to monitor the quality of the gas at the outlet of the rig. The bubblers that humidify the gas sit inside the test chamber alongside the devices. This ensures that all of the pipework and devices are at the same temperature, which prevents condensation from forming and affecting the results. The flow controllers that are used to make up the test gas mixture are controlled by a proprietary piece of software developed in-house. This software iterates each flow controller through a series of set points, which allows us to run extended automated experiments that can supply a series of different gas concentrations to the devices under test. A three-way solenoid valve and flow meter setup allows us to divert the gas flow to a flow meter at the exhaust. This is an in-process leak check that helps us to ensure that there are no leaks in the system and that the devices are seeing the desired flow rate and gas concentrations. In this slide, we present a picture of the rig parts that are internal to the environmental test chamber. This picture demonstrates the loading mechanism of the devices under test as they are placed in a bespoke loading jig so that the outlet of the synthetic breath rig passes through the device. As well as this, we can see the position of the bubblers and the environmental gas and monitoring sensors. By increasing the total flow rate, we can have 4 devices at the outlet of the rig. The outlet flow is divided evenly between the devices, which allows us to carry out experiments testing multiple devices simultaneously. This was extremely useful when building a matrix of training data, as it meant we could examine the behavior of multiple sensors when exposed to the same gas mixture at the same time. The jigs at the outlet of the rig are fully bespoke and removable. This gives us leverage in the design of alternative products, as we can just remove these jigs and add new ones to pass test gases mixtures through. As well as this, the versatility of the rig means that we can change the test gas mixtures completely, allowing us to examine the behavior of a myriad of different sensors and a wide range of different on-breath VOCs. The last thing I'll say about the rig before handing over to Riccardo is to detail some of the validation work that we have carried out to ensure that the OMED Health Breath Analyzer is trained under optimal circumstances. The initial version of the rig was designed in line with our set of requirements for the experiments. However, we found over time that there were many different modifications to the rig that needed to be made in order to improve experimental stability and performance. This led to a continuous improvement process over several months of experiments, which eventually led us to where we are today. There were several different learning curves that needed to be overcome, but I'll describe just 2 of them in this slide. So it's known that metal oxide semiconductors behave differently in environments of different oxygen concentrations. When detecting the presence of a reducing agent, reduction reactions occur on the surface of the metal oxide semiconductor, and the resistance to it is reduced by the supply of electrons. This is the fundamental mechanism by which metal oxide sensors are able to detect gases. When the environmental oxygen concentration varies, research has shown that the baseline of a sensor can be affected by changing the morphology of the surface oxide layer. Therefore, we decided that when building the training data matrix, we must ensure that the conditions of the sensors see match exhaled breath as closely as possible. As well as this, to simulate a user exhaling into the device, then putting it down in ambient oxygen, we typically flush the device with a 21% oxygen mixture after an exposure to the desired concentrations. Now, having said that, prior to a flush, what we wanted to do was ensure that we didn't clean the sensors down before they reached peak response. This brings me on to my second point. We found that during a 20-second exposure, which is the duration people are asked to give a breath sample into the OMED device, the sensor response will start to climb but continue climbing after the source of gas is removed. Now, this happens because the test gas mixture is still passing through the protective diffusion barrier, and it occurs in a real breath sample as well. To ensure that we were able to get the device to reach a peak, a three-way solenoid valve was utilised at the rig outlet to divert the gas flow immediately after a 20-second period. The devices are left in situ to allow for a plateau to be reached, and then they're cleaned down with a flush of air afterwards. This emulates a true breath sample as closely as possible. All of this is to say that there was a significant amount of validation work that was carried out as part of the development of this rig, which led to a robust system prior to the collection of the training data that was used to build the machine learning model. And I will now hand you over to Riccardo, who will talk you through the details of the collection and processing of this training data.
Thanks, Josh. Hi, everyone. I'm Riccardo, and I'm a senior data scientist here at Owlstone. My team and I are the ones who developed the machine learning algorithm that sits at the heart of the OMED device and interprets multiple sensor readings to return the concentration of hydrogen and methane on breath. To gather enough data to train the algorithm, we use the synthetic breath rig to generate a high amount of synthetic exhalations, which we call exposures, at different combinations of hydrogen and methane concentrations, at different temperatures and different humidities. The distribution of the gases concentrations were built to resemble what we expect the real distribution in the population to be, to maximize the representativeness of the training datasets. These are modeled around gamma distributions with a higher density of concentrations at the lower end of the range and lower density at the higher end. Some grid points at 25, 50, 75, and 100 parts per million were collected with increased frequency, which can be seen in the histograms. In total, we acquired 941 exposures with 4 different model devices for a total of over 3,700 data points, which form the basis of the training dataset for the algorithm, which would then learn the complex relationships between multiple sensor readings and the concentration of gases on breath. This dataset was then split into training set proper, validation, and test. The idea here is for the algorithm to learn on a randomly sampled subset of data, which is roughly 60% of all the data we got, with the validation set used in parallel throughout the training process to check for model overfitting. The way this looks like is as the algorithm learns, we'll see performance improve on both the training and the validation sets. When at some point performance will inevitably stop improving on the validation set, any improvement on the training set is considered model overfitting and assigns the learning should be stopped. At the end of this process, the model is finally evaluated on a test set that it has never seen before. And performance on this set is taken as a metric of how generalizable are the results, or in other words, how the model is expected to perform on truly new data. The graphs here show comparison of true concentrations on the x-axis and the model predicted concentrations on the y-axis. So minimal spread around the green line means good predictive capabilities. Performance on the test set being virtually equivalent to performance on the train set means the algorithm is likely to generalize well to new data. All in all, we are in a good spot for predicting hydrogen and methane concentrations on synthetic breath. But we don't want to just do that, read gas concentrations on synthetic breath. We want to be able to do that also in real human breath. In order to test the device capabilities to quantify hydrogen and methane on real human breath, we ran a small experiment which paired OMED device readings with a standard benchtop hydrogen and methane breath test, or HMBT. Specifically, we had a total of 54 participants giving 243 breath sample sessions. which every session consisted in an initial HMBT sample, followed by an OMED device sample, and again an HMBT sample, at intervals of 3 minutes. This way, we could use the average of the two HMBT samples as our ground truth, or the true levels of hydrogen and methane in the participants' breath, against which we could compare the OMED device reading and its accuracy. The scatter plots are similar to the ones on the previous slide, with the green line highlighting perfect ideal accuracy. On the left of the panel, we have performance of OMED against the average of the two HMBT readings. And additionally, the overall error distribution is shown in the graphs on the right and was obtained by applying Gaussian kernel density estimation on the model residuals. And as a terms of comparison, the same has been done to error between the first and second measurements with the HMBT, showing the HMBT error against itself. In the line plots, this is shown as the dashed lines. The error distributions look comparable between OMED and the HMBT, which is a very promising result. But how accurate is accurate enough? To answer this question, we needed to establish how well the OMED device's accuracy would hold up in a diagnostic setting. To do so, we ran a separate small study in collaboration with an external partner, in which we provided real users looking to test for SIBO and IMO with an OMED device, together with sampling equipment for gas chromatography, HMBT, which is the gold standard for hydrogen and methane testing. SIBO stands for small intestine bacterial overgrowth, and IMO stands for intestinal methanogen overgrowth, and both are pathologies rooted in microbial overgrowth in the GI tract. The test works like this. The patient is asked to follow a specific diet beforehand and fast overnight. Then a baseline reading is taken, after which a substrate is ingested, which is usually lactulose. And then 9 additional readings are taken at intervals of 15 minutes. The diagnostic criterion for SIBO in the EU is the presence of any hydrogen reading higher than 10 parts per million over baseline within 60 minutes of the ingestion of the substrate. The IMO diagnostic criterion is the presence of any methane reading above 10 parts per million at any point during the test. The aim of the study was to compare the diagnosis that would be made based on applying SIBO and IMO diagnostic criteria to the results on the HMBT samples and the OMED readings. The confusion matrices shown here are counts of the tests that had the same or different diagnostic outcome between the HMBT and OMED. On the diagonal, we have tests that were negative for both methods on the top left, and tests that were positive for both methods on the bottom right. Bottom left are false negatives, and top right are false positives. From these results, we can calculate that OMED is more than 90% concordant with the gold standard HMBT when making SIBO and IMO diagnosis. We are planning to replicate these results in a separate, larger study very soon. And last, the development of the OMED device would not have been possible without the support of existing knowledge on SIBO and IMO. Knowing the right biomarkers and the range of concentrations these biomarkers present on breath in healthy individuals and individuals affected by pathologies was absolutely crucial in specifying appropriate product requirements in terms of both sensitivity and specificity to certain compounds and accuracy of the concentration measurements. Instruments like the Owlstone VOC Atlas, by providing information and data about relationships between diseases and specific VOCs found on breath, together with the precise levels those compounds are expected to be present on breath, can be a cornerstone for the development of analytical devices like it was for the OMED device. Thank you for your attention.
Excellent. We've got a few questions here. One relates to the MOS sensors. So MOS sensors may be sensitive to different VOCs. How do you deal with this? Maybe Josh, you can talk about the filtering capability about the MOS sensors there.
Yeah, so within the device, we utilize a diffusion membrane, which is essentially a protective barrier which we place over both of the sensors. This helps to prevent any of the other on-breath VOCs that the sensors could be cross-sensitive to from inducing a response in them. We've demonstrated experimentally across a range of different typical on-breath VOCs that we would see that the filter membrane is capable of removing any effect of those.
Excellent, thank you. Next question here was, why did you use the threshold bottles to humidify the gas and not another method like CEM? I'm not sure what CEM is, so hopefully you do.
Yeah, no, that's good. Yeah, that's a good question. So CEM would be a controlled evaporator mixture. So it would be a way of essentially demonstrating, generating a vapor. It would have worked. It would have equally been a good method. The reason we decided against it was because we wanted to ensure that we could equally control the environmental conditions that the sensors were in alongside the breath. So using the Dreschel bottles and the current method of humidification that we have, it meant that all of the internal tubing and the devices were at the same temperature, which helped to prevent condensation from forming. Were we to use a CEM or a different method of vaporizing, we would probably have to insulate the tubing and ensure that everything is held at the same temperature to prevent condensation from forming. So it's a way around that, essentially.
Excellent, thanks. Question for Rick. There's two sensors. You have some cross-reactivity from one sensor to the hydrogen methane. I was wondering if you could elaborate a little bit further on how that is dealt with in the classifier model that goes from the raw sensor output to the ppm outputs.
Yes, that's a good question. I think it stems from the fact that we do not have easy access to methane-specific metal oxide sensor. So the one that we got responds to both. That's why we need the other. So the model can make sense of the signal coming from both because we know that one signal is proportional to hydrogen. So that is used to remove, in a way, the proportion of the signal that is depending on hydrogen on the second sensor, leaving us with the pure methane signal. Does it answer the question?
Yep. Next question. Great work. This could serve as a model for developing various point-of-care and home novel diagnostic systems. How is the unit and sensors scrubbed to remove any contaminant VOCs for reuse? Maybe Josh, you can pick that up because I suspect that relates again back to the scrubber that's in the unit.
It would, yes. So regarding that, any devices that are used on the rig, we utilize a set of activated carbon scrubbers in line prior to the devices to ensure that any contaminants and such don't pass on to the devices. If we're talking about, say, for example, were the devices to be used by people for breath sample afterwards, the mouthpieces are cleaned separately and placed on the devices afterwards. So everything is essentially cleaned afterwards.
I think the question might relate more to when a user has a device. So between measurements, is there any
I see.
or hang up? Yeah.
I see. Sorry about that. Yeah, no, that makes sense. So essentially, over time, the devices themselves, we've demonstrated that any contaminants or any hydrogen or methane diffuses off over time. We found that there's about a 15-minute limit by which is imposed in the device itself, whereby it takes that long for it to reach another stable baseline so that another sample can be given. We also encourage people when they're using it to charge it as often as possible, because with metal oxide sensors, being switched on helps to burn off any other contaminants that are on there, and the sensors are usually switched on whilst the device is in charge mode.
Got it. Thank you. And next question, maybe for Rick. How does the device know when it's being breathed into? Is there a button or a user control?
Yeah. So relying on user input is tempting, but may then result in inaccuracy, basically. So instead of trusting the user, we have developed an algorithm that detects when the device is being breathed into. The algorithm, like after the warm up and the device is ready to take a sample, the algorithm continuously looks for a signal. Then we know it coincides with the start of breath. We also have an algorithm that detects the end of a breath, so that we know that the user has exhaled for an appropriate amount of time, so that we only process valid samples.
Excellent, thank you. Next question for Josh. This relates to the synthetic breath rig, I think. So how do you add other on-breath VOCs to a gas mixture?
So yeah, this is a great question. So we utilized this in the demonstration of the capability of the filter stack. So in generalized training data experiments that we were discussing as part of the presentation, we decided against adding any VOCs to that. And the reason for that was because we were able to demonstrate that we were able to remove the effects of the on-breath VOCs using the filter membrane, which sits over the sensors. So in order to do that, we used a dynamic headspace sampler with concentrations of typical on-breath VOCs, i.e. acetone, isoprene, etc., which were actually, in fact, orders of magnitude higher than what you would typically expect to see on breath. And the baselines of the sensors weren't perturbed at all. So that gave us a lot of confidence that the filter membrane is removing the effects of other on-breath VOCs.
Excellent, thanks. We have time for one or two quick questions. Maybe this one for Rick. What's the limit of quantification of the device? Is it ppm or ppb levels?
It's definitely in the pp parts per million range. I do not have the exact quantification of the exact calculations for the limit of quantification, but it's definitely in the parts per million range.
Yeah, and I suppose that's one of the things that's quite special about hydrogen and methane. They do tend to be much higher abundance than other compounds. So
yes.
Things about translatability of VOCs or target gases to small, low-cost devices, hydrogen methane are quite special in that regard. Okay, maybe this one for Josh. Can you comment on the stability of readings over time and the lifetime of the device when the user has it?
Yeah, so the original requirements of the device were a lifetime of 2 years with stable, repeatable readings within that. What we've demonstrated as part of the training data collect was that the number of exposures, well, in fact, the number of exposures that Rick mentioned during the training data collect actually exceeded the number that we would expect a device to see in its lifetime, and though the devices that were used in the training data collects demonstrated good repeatable stability throughout that timeline, so that gives us that confidence. And we have other experiments ongoing to look into device stability over time, but we have a lot of confidence in that is very good.
Thanks again both for taking the time to present the work and answer the questions.